Hey, hi-fi everyone. This is the next video of my channel and welcome to my channel. In this video, we will discuss about the antigen and antibodies. So let's get started. Antigen and antibodies. This is the part 1 video, and in this part we will cover the antigen only. So, what is the antigen? Antigen is a substance that satisfies two distinct immunological properties. Immunological properties, which are that? First is the immunogenicity, second is antigenicity. So, what is immunogenicity? Immunogenicity is the ability of an antigen to induce an immune response in the body. And antigenicity is the ability of the antigen to combine with the final products, antibodies or the T surface receptors. The small unit of the antigen is called as the epitope. So, epitope is the smallest unit of the antigenicity. So this is all about the definition of antigenicity or antigens. So what are the factors which affects or influence the immunogenicity? There are 11 to 12 factors which affects the immunogenicity. Let's discuss about them. First is the size of the antigen. The size of the antigen really matters. For example, as the largest the size, more important is molecule as immunogen. That means the largest the size, the immunogenicity will be more. Second is chemical nature of antigen. As you all know, the antigen is the proteins which have higher antigenic properties as compared to the carbohydrates. Okay, and the antigens which are carbohydrates have more antigenic property as compared to the lipids and nucleic acid. The antigens which is comprises of lipids have the more antigenic property than the nucleic acids. So third point is the susceptibility. So susceptibility of an antigen to the tissue enzyme is really matters. How? Let's discuss. Only substances that are susceptible to the action of tissue enzymes are immunogenic. Fourth one is the structural complexity. Simple homopolymers made up of a single amino acid which lacks immunogenicity. That means the antigens which is the homopolymers and made up of a single amino acid does not have the immunogenicity, and the polymer antigens which is made up of two or more amino acids are very immunogenic in nature. Fifth one is the foreignness to the host. Only factor which determines the immunogenicity, hyper, is the phylogenetic distance between the antigen and host is more the immunogenicity. Genetic factor is the sixth one, and it is also very important, and genetic factor depends on the individuals to individuals. For example, different individuals of a given species shows different types of immune responses toward the same antigen. Optimal dose of an antigen is also a significant point. An antigen is immunologically active only in the optimal dose range, and if it is provided more than the optimal dose or the optimal range, then the immunogenicity will be altered. Eighth one is the route of antigen administration. For example, immune response is better induced if it is followed by the parental pathway of infection may influence immunogenicity. Repeated doses of antigens is also significant. Repeated doses of an antigen over a period of time are needed to generate an adequate immune response. This is due to that fact that the role of memory cell in the secondary immune response is activated. However, after certain doses of antigen, no further increase in antibody response is seen. So, the repeated dose of antigen should be given with the precautions. Multiple antigens. If two or more antigens are given simultaneously, the effects vary, and these effects vary from person to person and depends on the above 9 factors discussed. 11th is the adjuvant. Any substance that enhances immunogenicity of an antigen which is added to the vaccine to increase the immunogenicity of the vaccine. Okay, so this is all about the 11th factor which affects the immunogenicity, and now we will discuss about biological classes of the antigen. What are the biological classes of the antigen? So, biological classification of the antigen can be divided into the two parts. The T cell dependent antigens, the T cell independent antigens. So let's discuss T cell dependent antigens first. So T cell dependent antigens activates the cell which secretes cytokines that in turn stimulates the B cell to produce the antibodies. As you all know that B cells produces the antibodies, and T cell dependent antigens, what they do? They activates the cell which secretes the cytokinins, and cytokinins in turn induces the B cell and they produces the antibodies. They are structurally complex protein in nature, and T cell dependent antigens are immunogenic over a period of dose. They have memory, antigen processing is needed, rapidly metabolized, active the mature B cells only. So these are the characteristics of the T cell dependent antigens. Now, T cell independent antigens. What are they? They are structures in a simple lipids. So, they are simple lipids. They are dose dependent immunogenicity. They have no memory and activate both the mature and immature B cells. They directly bind to the Ig receptors present on the B cell and stimulates the B cells polyclonal. Okay, so this is all about the antigens. We have discussed the antigens introduction, the factors influence the immunogenicity, and biological classification of the antigens. If you find this video helpful, please like, share, and subscribe. Thanks for